Aloha everyone, and welcome to today's lecture on Chapter 14 in your e-text. So in Chapter 13 we talked all about evolution and natural selection and how over time organisms tend to adapt to their environment, and that is this process of natural selection. And then when we actually see a change in the alleles, that's when we have evolution occurring. Today we're going to talk about something a little bit different. We're going to focus on non-adaptive evolution and speciation. So in other words, evolution that does not occur due to adapting to the environment. For our learning objectives for Chapter 14, first of all, we're going to learn how different evolutionary mechanisms influence the composition of a gene pool. And if you don't know what a gene pool is yet, don't worry, we're going to talk about what exactly a gene pool is as well. Then we're going to look at understanding how the gene pool of evolving population compares to the gene pool of a non-evolving population. So in this lecture, we're going to be looking at things such as gene flow, and we're going to be talking a lot about genetic drift and some different types of genetic drift, such as the bottleneck effect and the founder effect. Then for our third learning objective, we're going to talk about what exactly is a species, and we're going to focus specifically on the biological concept of a species because here we are in a biology course, right? And then we're also going to learn how new species are able to come about through the process of speciation. So let's go ahead and get started. Your e-text focuses a lot on this topic of population genetics. So population genetics is the study of the genetic makeup of populations and how that genetic composition changes. In other words, it's kind of understanding how a group of organisms is going to cope or deal with environmental changes. So with population genetics, we're really studying many different things. But mostly we are relying on DNA, right? Because DNA is that genetic code. So population genetics, it makes sense that we're relying a lot on the DNA. And we're looking at how that DNA is changing across a population over time. And we're asking questions about, whether a population is evolving. If it is evolving, how is it evolving? In what ways is it evolving? So with population genetics, we're looking at entire populations because remember what we learned in our last lecture, only populations evolve, not individuals. So when looking at a population's genetics, we're really looking at what we call their gene pool. The gene pool is the total collection of alleles in a population. So scientists are able to use DNA sequences to study these alleles in the gene pool. And if you've read the e-text, you know this chapter kind of focuses on these scientists in New York City, kind of the Manhattan area, and they're studying mice populations, which I'm going to be honest, mice are the only organism I'm afraid of. So I really went back and forth as to whether I was actually going to use the examples from the book for this lecture, but we do go through, like a little bit later, there's going to be a difficult concept with the Hardy-Weinberg equation. So I did decide to keep the examples the same just because anytime it's a difficult topic, I try to keep it the same as the textbook so you have, double the amount of resources there. So I'm explaining what's in the textbook, and if that doesn't make sense, just going back and forth tends to help students sometimes. So this chapter is all about these mice populations in New York City. And when we look at the gene pools, this would be considered a gene pool. This is that original population, and we're looking at the collection of alleles in this population. So each mouse has 2 alleles for each one of their genes, just like we as humans, right? So if there are 15 mice, that means 15 times 2 gives us a total of 30 alleles in the population for each one of the genes. And this tells you that in the beginning, you have different colors. You have blue, red, and yellow. So in the beginning, 6 out of 30 alleles are blue, so that's 20%. 12 out of 30 are red, which is 40%. And 12 out of 30 are yellow, which is also 40%. And if you add them up, 20 plus 40 plus 40, that gives us 100% of the alleles in this particular gene pool. So we have the starting point for a gene pool. Then over time, we can go back to that gene pool and say, hey, are things changing? Is that population evolving? So we wait several generations and we go back to this gene pool. And we can look again at these allele frequencies. And if we see a change in the allele frequencies, that means some type of evolution has occurred, right? Because remember, the definition for evolution is a change in those allele frequencies over time. So now we have 12 out of 30, out of 38, because now we have a few more mice here, right? 12 out of 38 gives us 32% for blue, 21% for red, and for yellow we have 47%. So are these percentages exactly the same? No, right? We see an increase in the blue allele, and we see a decrease, almost half, decreased by half, of this red allele. So a change in the allele frequency means that evolution has occurred in that population in that particular gene pool. The genetic diversity of a gene pool is really critical for the survival of populations. So scientists like to look at the allele frequencies to see, hey, are they changing over time? And if they are changing, that means obviously a population is evolving, right? So the allele frequency, just to kind of define this for you, this is the relative proportion of an allele in a population. So again, evolution of populations is caused by a change in this allele frequency over time. For these changes in the alleles, they can be many different things. So this change can be positive. This change can be neutral, meaning it really doesn't seem to affect the population, or it can actually be negative for the population in terms of the fitness. And remember, we defined that term fitness in an evolutionary sense. It means the ability of an organism to survive and then pass its genetic material on to the next generation. So this can result in that population becoming more adapted to its environment. So in other words, as we learned last chapter as well, natural selection results in a population adapting, right? So this kind of goes along with what we learned last chapter. And again, these changes aren't necessarily always going to be good, but they're generally going to result in some type of adaptation. So what exactly causes these changes in allele frequency? We looked at the definition of allele frequency. We know that a change in allele frequency leads to evolution, but what really causes this change? Well, one of these is something we talked about last chapter, and that is natural selection. Natural selection occurs when the population becomes better adapted. We already talked about this in depth last lecture, so we know we have natural selection. This is what we call adaptive evolution because we're becoming better adapted. So I wish I would have wrote this on here, but natural selection, evolution that results as that population becoming better adapted is adaptive evolution. And we talked about this last lecture. But you probably noticed from the title of this lecture that we're not going to be focusing on natural selection and adaptive evolution. This chapter is going to be all about non-adaptive evolution, and so that's what we're going to focus on for the rest of this lecture. So non-adaptive evolution is where things don't really make as much sense. We have these random changes that do not really affect the fitness for an environment. So we're not really adapting to the environment, which is why we call it non-adaptive. These can be caused by various things. So by mutation, and this should say not genetic drive, but genetic drift, and gene flow. A lot of times with non-adaptive evolution, we're looking at that DNA to try to figure out what exactly happened. It allows us to see that some type of evolution is occurring because, again, evolution is a change in that allele frequency, so we have to focus on that DNA. So in the following slides and really the rest of this lecture, we are going to be focusing on non-adaptive evolution. One of the ways that we see non-adaptive evolution occurring is through a process known as genetic drift. Genetic drift is where we have a random change in the allele frequencies between generations, and this occurs purely by chance. So what this means is that we don't really have an adaptation occurring. It's just random, it's by chance that we're seeing this evolution occur through genetic drift. In genetic drift, what we see is that a subset of that population is going to reproduce. So in other words, not the whole population reproduces. It can be for various reasons, but just part of that population reproduces. And that means that because only part of it is reproducing, we have just a subset of the alleles that are represented in the next generation. This can decrease the genetic diversity of a population. So what this means is we really don't have as many alleles represented in the population because we aren't passing all of them on to the next generation. Something else we see with genetic drift is that the smaller the population, the greater the effect of genetic drift, right? Think of this really simply. If we have 1 million organisms and just a subset of them reproduces, so say 800,000, we still have a pretty good amount of genetic diversity, right? But if we have a population of 10 and only 5 reproduce, then we're going to have a really big decrease in that genetic diversity, right? So the smaller the population, the greater the effect of genetic drift. Genetic drift tends to have much more dramatic effects in these smaller populations because in a population with fewer individuals, any individual that does not reproduce could really spell the complete loss of alleles from that population. So having more of a diverse gene pool gives the population much more flexibility to survive in a changing environment. The more genetically diverse a population, the more ways it has in order to adapt, right? So if you think about even going back to like the peppered moths, if the peppered moths only had white alleles available and that suit occurred, they wouldn't be able to change their coloration, right? So the more diverse a population is, the better capable it is. You want more alleles available in that population. Looking at one example of genetic drift. So an example could be, say, a population gets separated from another population. And this is an example of genetic drift that's shown in your textbook. And it shows what can happen when populations get separated. So if you look here at these big red dots, right? Each one of those big red dots are the various study sites in New York City. I think there are 15 of them total. Yep, 15 of these big red dots. And the different shades of green, so you can see this dark green, lighter green, these different shades show you the percent canopy cover. And you can see that urbanization has really fragmented the green spaces into very distinct patches, right? So you have just these distinct patches of green now, right? Whereas back in the day, this used to be completely green. So what I'm meaning in terms of urbanization in New York City, I don't know if you all have been there. I have been there a couple of times. There are all these huge skyscrapers, a lot of buildings. This has really cut down on the landscape. So there are a lot of different populations of wild organisms that have been separated by all the urban buildings in New York City. Some of these patches that you see are connected by what we term greenways, and some are not. So these greenways would be kind of these really skinny patches that you can see, like these skinny bits of green that lead to another patch. However, there are some patches that are not connected at all, right? So if you look at 13 right here, this patch right here is not connected to any of those other study sites. So what we find is that there tend to be different populations, meaning different alleles, in these patches that are not connected via greenways to other patches. So that's kind of this map here kind of showing you how some of those patches are connected by greenways and some are not. And if they're not connected, we see different alleles. So over here is the data that's collected from the DNA. So what they did is they clipped the end of the mouse's tails, which does not actually hurt the mouse, and then they studied the DNA at all of these 15 different study sites. So each bar here is one mouse's genetics. So researchers collected that DNA from 312 mice at these 15 study locations. They analyzed each one of those mouses' DNA to get a better sense of how related the populations were. They assigned the mice with similar genotypes particular colors and sorted them by location. And they found that mice within a population shared more alleles with others than they did with mice from other populations. So in other words, these populations are kind of becoming distinct because they cannot connect via greenways with other populations. So each one of these vertical bars shows the data of one mouse. Mice sharing similar genetic sequences are shown in the same color, so all those in green, all those in yellow, all those in pink. And mice in one population share more colors, meaning more alleles, with each other than they do with mice in other populations. So definitely a really cool study that your textbook went over. So there are similar genetics based on where the mice are from. So they're breeding within their green areas and they are not mixing. Genetic drift can occur from more things than just the populations getting separated by urban buildings, right? It can also be the result of flooding. It can be the result of natural disasters. Many different things can result in genetic drift. Pretty much anything that separates an original population. Again, genetic drift tends to be more dramatic in smaller populations because in a population with fewer individuals, any individual that does not produce can lead to a loss of alleles from that population. Another example of genetic drift is what we term a bottleneck effect. So a bottleneck effect is a type of genetic drift that occurs when a population is suddenly reduced to a small number of individuals. In other words, you're creating this population bottleneck. Only a few individuals are able to get through that bottleneck and survive and reproduce. So a bottleneck effect like rapid habitat loss eliminates a large percentage of the original population. So again, going back to the mice in the e-text, by chance, the mice that actually survive urbanization are going to have a reduced allele frequency than the original population. So you can see that here. You have your allele frequencies before this genetic drift, before this bottleneck effect. You have this bottleneck effect, like in this example, rapid habitat loss. That eliminates a very large percentage of the population. So you only have a few surviving individuals. They are less diverse afterwards, right? You've completely lost that red allele, right? So less diverse population is your end result here. So the bottleneck effect is again a type of genetic drift, and this occurs when that population is suddenly reduced to a small number of individuals, and as a result, alleles are lost from the populations. So taking it outside of the e-text, a very good example of genetic drift is seen in cheetahs. So cheetahs have had a significant loss of genetic diversity within their population, which is believed to be caused by a historical population bottleneck, where a small number of individuals were left to reproduce, leading to a reduced gene pool and increased inbreeding, which we'll talk about inbreeding later. But this made the cheetahs very genetically similar to each other, with limited ability to really adapt to environmental changes because their gene pool was so small. And they are pretty much almost genetic clones to one another due to this drift. So there have actually been some articles posted about you can take a skin graft from one cheetah to another cheetah and it works. There's not rejection of it because their DNA is so close. Whereas if you were to take a skin graft from one human to another and you didn't do any blood typing or anything like that, you're probably going to see rejection, right? But that's not the case with cheetahs because they are so genetically similar. Another type of genetic drift is what we term the founder effect. So this is a type of genetic drift in which a small number of individuals leaves one population. So it's not that just a small amount survives or they get separated. It's a small amount leaves one population and establishes a new one. You could even think of that as like colonization of America, right? A small subset of people left Europe, came over here to colonize America. So the founder effect is a type of genetic drift in which a small number of individuals, again, leave one population and they establish a new population elsewhere. So by chance, that newly established population can have a lower genetic diversity than the original population because you have less alleles, right? So the genetic diversity of the new population reflects the reduced diversity of the founders. So they have less diversity than that much bigger original population. So the example from the e-text is where you have this large, diverse original population in New York City before urbanization occurred, right? And then you see a few of these mice are like, oh no, this is, buildings are coming up, we need to find a new home. So maybe they go to the outskirts. Maybe they go to Central Park. Maybe they go to some place that just seems like a better home for them. And they establish their own population. You can see they're much less diverse than that original population, right? We've completely lost the blue allele in this case. We also see numerous founder effects that are created for different species of fruit fly right here on the Hawaiian Islands. So studies of the genomes of these flies have revealed that a given species is often more closely related to species on a neighboring island than it is to species on its own island. So this suggests that species on the same island were actually created by separate founder events. So they are genetically closer to their parent population than to the founded populations, which may be geographically closer. So for example, the different fruit fly species that we see on the island of Oahu, they are more different from one another on this island than they are from, say, on Maui or from the Big Island. So definitely very interesting, and again, this is a result of founder effects because that's where they're closer to that founding population than from a population that comes from a different founding population, even though they're technically on the same island. So again, looking at genetic drift, now we've looked at a couple different examples of it with the bottleneck effect and the founder effect. So genetic drift is able to reintroduce genetic diversity by bringing in new alleles, mutation, and via gene flow. So what exactly is gene flow? Well, gene flow is the movement of alleles from one population to another. So really just like what it sounds, those alleles are moving, right? So in this figure, you can see these two populations. Population A is red and yellow alleles of these mice. Population B is blue and yellow alleles of these mice. And what's occurring here is you have random migration and interbreeding, so breeding between the two populations. And this actually introduces red alleles into population B, which before just had blue and yellow alleles. So again, this gene flow is just the movement of the alleles from one population to another, right? We now introduced this red allele into population B. Right? So that is how your gene flow is working. We've talked before in a previous lecture about mutation, right? Mutation generally occurs through that process of DNA replication, right? We try to catch the mistakes, but we don't always catch them in time, and so over time we tend to see some mutations occurring, and that can increase genetic diversity as well. You have probably heard of inbreeding before. So urbanization can actually prevent gene flow. So what we just talked about on that last slide, where you're going from one population to another, say you put a big brick building in the middle of that path between the two populations. That's going to prevent that gene flow, right? And it can lead to a lack of genetic diversity, which when you don't have DNA that is distinct enough, that results in what we term inbreeding. Inbreeding is mating between closely related individuals. So it's as simple as, say, like in the human population, if cousins were to mate or siblings were to mate, that would be inbreeding, right? Your DNA is too similar. You should not be reproducing. This does not change the allele frequency within the population. So we talked about genetic drift and how we see that decrease in the genetic diversity. We don't see really a change in allele frequency here with inbreeding, but it does increase the proportion of homozygous individuals to the heterozygotes. So remember, homozygous individuals are those that have the same alleles, either the homozygous dominant or homozygous recessive. So what we're doing is we're passing down the same alleles, so the odds of having heterozygotes that have one of each allele are much slimmer, and we're much less likely to have heterozygotes. With inbreeding, we can get what is termed an inbreeding depression. So this is where closely related individuals are more likely to share the same alleles. Negative reproductive consequences for a population are going to result from this inbreeding depression. And this is generally going to be associated with a high frequency of homozygous individuals possessing harmful recessive alleles. So in other words, most of the individuals are going to be homozygous recessive. And if you remember last unit, we talked about a lot of conditions that can be really harmful are homozygous recessive conditions. So we see a lot more of these conditions in these inbreeding populations. So we have an increased chance of two recessive alleles coming together during mating. And when that happens, the homozygous recessive genotypes are created, and the previously hidden recessive alleles, so those that were just carriers before, now those recessive alleles start to affect the phenotypes. So a couple different examples of this. This is kind of, I think, a little extreme of a picture, but you may have heard of the blue people of Kentucky. So this is a one example of an inbreeding depression. So this family has passed down this rare genetic blood condition that turns the skin blue due to a lack of oxygen in their blood. Another example, one that your e-text talks a little bit about, is polydactyly, so having more than 5 fingers on each hand. And this is sometimes seen in Amish people because they're breeding within their population, right? And so we can see, right, more than 5 fingers on each hand. That's an example of polydactyly as well. So polydactyly in the Amish and the blue people of Kentucky are 2 examples of inbreeding depressions. Something your book talks a little bit about is the effects of urbanization. And researchers actually compared the DNA between city mice and country mice. And they found that country mice and urban mice or city mice actually differ in the genes associated with immunity and their response to stress. And these genes may be responding to selective pressures in the urban environment, such as, say, exposure to pollution and competition for food and mates. So it's probably because these have been selected for again and again in these urban mice to help them cope with the stresses of city living. And some of these stresses include, right, the influx of humans. There's a lot more noise in the city than there is in the country. There's also a lot more diseases, right? So they are probably living in a much more toxic environment in the city than in the country. There's more toxic chemicals and a lot more pollutants. And because these alleles have been selected for again and again, they've been passed down from generation to generation, such that we're now actually able to see a difference in these alleles for the genes that are associated with the response to stress and immunity. So kind of a cool study that your e-text talked a little bit about, where we can actually see a change in the alleles for these country versus the urban mice. You'll probably remember that I've said in the past that I don't like to put tables on slides. Well, this is an exception, right? This is a really important table, and it's something you really need to memorize. I would suggest making flashcards. So make a flashcard for natural selection, for mutation, genetic drift, gene flow. And you want to know how those allele frequencies change. Definitely focus on adaptive versus non-adaptive. So the only adaptive one is natural selection, right? And then I also want you to know whether genetic diversity is affected. So for natural selection, usually it decreases. For mutation, it increases, right? Because you're creating a new genetic makeup. Genetic drift, we talked about how it usually decreases. And then gene flow is obviously increasing, right? Because you're having that flow of alleles from one population to the next. So I highly encourage you, make 4 note cards. So we're just going to look at the example of genetic drift. Put genetic drift on the front. On the back of that note card, put that you're seeing alleles change due to chance, it's non-adaptive, and genetic diversity decreases. Or you can make multiple note cards, like how does genetic drift affect genetic diversity? And then on the back, put it decreases it. Things like that. Make sure you know everything in this table. Something else I want to point out is that it's important to remember that evolution is pretty much happening all of the time. And all 4 of these forms of evolution are happening at the same time. It's not that, okay, let's spend a couple months with natural selection, then we're going to move to genetic drift, then we'll see some mutation. No, it's all occurring simultaneously. Now we're going to get into what is probably the most difficult and definitely most math-heavy portion of this chapter. So most of the time when we are studying population genetics and dealing with long-lived organisms, it's really impossible to see evolution occurring in a lifetime. It's also really hard to get a good representative sample of a population because sometimes there are just so many individuals. So what we use to kind of get an idea of what's occurring for a baseline is what we term the Hardy-Weinberg equilibrium. And again, we use this to get a baseline for the populations. Hardy-Weinberg equilibrium is pretty much impossible to have in nature. So Hardy-Weinberg states that in a non-evolving population, the allele and genotype frequencies do not change over time. So the Hardy-Weinberg equilibrium is just used to identify genes that have changed because of evolutionary mechanisms. And again, this is really impossible to have in nature. We're not going to see this Hardy-Weinberg equilibrium. It's just kind of this pretend situation that we use to get a baseline for the condition of an environment. It's to get a baseline for the populations. So again, Hardy-Weinberg is used as a baseline to judge if a population is evolving. So in other words, we have that baseline condition of, hey, nothing is evolving, nothing is changing. And then if we have that baseline, we can compare it to the actual situation to be able to say, yes, evolution is occurring, or no, it is not occurring. So now for the equation. Are you ready for the math part of this? I promise it's not too scary. So the Hardy-Weinberg equation states that p squared plus 2pq plus q squared equals 1. You'll also see this written sometimes as p plus q equals 1, where p squared is the frequency of the homozygous dominants, 2pq is the frequency of the heterozygotes, and q squared is the frequency of homozygous recessive. So the Hardy-Weinberg equation is essentially this mathematical formula that relates the allele frequencies, right? Because homozygous dominants, heterozygotes, and homozygous recessive are your three possible types of alleles, right? So it relates the allele frequencies to the genotype frequencies in a population at Hardy-Weinberg equilibrium. So for Hardy-Weinberg equilibrium, we must have 5 necessary conditions. And again, this is pretty much impossible because we really have no say over mutation. Our DNA regularly mutates, and so do other organisms' DNA. So if it mutates and it's not caught in some checkpoint, then you have mutation. So this idea of no mutation creating new alleles, it's really hard to have that. It's pretty much impossible. Secondly, we must have no natural selection favoring some alleles over others. So in other words, this means this population is not adapting to the environment at all. Third, we need an infinitely large population size, and therefore no genetic drift because the population is so large that we're not seeing any effects from genetic drift. And this just really is not really possible. Fourth, no influx of alleles from neighboring populations. In other words, what we talked about earlier this lecture, we have no gene flow occurring, no migration and interbreeding back and forth. And fifth, we do not have random mating, we have random mating of individuals, which this really doesn't usually happen in real life, right? So these are the 5 conditions that we need to be in Hardy-Weinberg equilibrium. And I already mentioned multiple times, this is pretty much impossible, right? But again, even though this is impossible, it does give us a baseline to determine if a population is evolving. Okay, so looking at a figure kind of showing this Hardy-Weinberg equation. Again, this is just basically to give us a baseline to judge whether evolution is occurring in a population or not. So we can take this and say what we would expect if that population is not evolving. So then if we get something different for our allele frequencies, we know that, hey, that population is evolving. So what we do with this Hardy-Weinberg equation is we go ahead and plug in the frequency of different alleles into the equation. So if you know that 6 out of 10 mice have the red allele, then you would plug in 0.6, right? So 0.6 would be for this p squared. And if you know that 3 have the green allele, then you would plug in 0.3 because it's 30%, which percent you divide by 100, right? So that's how you get 0.3 instead of putting in 3. So you plug in 3 and then you can solve the equation, right? So you know if you know one of them, you can find out the others. And we're going to look at an example of this on the next slide. But again, this is happening in a non-evolving population. The mice will be mating randomly, no mutation and no natural selection, and no genetic drift. So when we add up the genotype frequencies in this non-evolving population, we get the Hardy-Weinberg equation. And again, we're going to look at an actual example on the next slide. Here we have 2 different colors of mice, right? So we have brown mice, which is going to be your dominant phenotype, And if that organism is homozygous recessive for the, or homozygous dominant for the genotype, so uppercase B, and if it is heterozygous, big B, little b, then that means you will have a brown mouse. If you have two lowercase b's, that mouse is homozygous recessive and it will be white in color. So if you look at the genotype frequencies, P squared would be your homozygous dominant, 2PQ would be your heterozygotes, and Q squared would be your homozygous recessive. And that all must add up to 1, or in other words, 100% of the population. So down here, we're kind of given an example. So we know that Q squared, so Q squared is going to be the frequency of homozygous recessives. We know that this is 9%, or 0.09. So if we take the square root of that, so that's Q squared, if we take the square root, we know that Q is 0.3. And as I mentioned, P plus Q equals 1. So we put this into an algebraic equation, P plus 0.3 equals 1, and then you're simply going to solve that. Excuse me. So we would take and move over 0.3, so it would be 1 minus 0.3 equals 0.7. And say it's been a long time since you took algebra. I would tell you, make sure you check your math on this, right? So you should be able to then take P plus Q equals 1. So we know Q is 0.3. We found that we think that P equals 0.7. Does 0.3 plus 0.7 equal 1? Yes, it does. So we know that is correct. And this is a little more complex than you're going to see on the exam or anything like that. But I felt like if I didn't go through an example, you would probably be like, okay, why in the world do we care about this Hardy-Weinberg equation? So I went through this example, and if you see examples like this on the exam, it's going to, tell you things like, hey, remember to use the Hardy-Weinberg equation. Hey, check your math, use basic algebra, things like that. And you'll be able to use your cell phone to plug in the numbers or a calculator if you have that. But I just wanted to kind of give you an example of how exactly we can use this. I do need you to memorize these equations. So memorize that P plus Q equals 1 and P squared plus 2PQ equals Q squared. And make sure that you know that P is your homozygous, P squared is your homozygous dominant, 2PQ is your heterozygous, and Q squared is your homozygous recessive. Now we're going to move on to our last learning objective, which is talking all about what exactly a species is and how new species can evolve. So a species is really a kind of living thing. And there are many different species concepts that can be used to define what a species is. The biological species concept is the most frequently used species concept. So the biological species concept states that a population of individuals whose members can interbreed and produce fertile offspring, if that is the case, then that is a species. So in other words, they have to be able to interbreed and actually produce fertile offspring. Different species cannot mate because they are reproductively isolated. And we will go over some different examples of what exactly reproductive isolation means and how it can actually happen to different species and different populations. So we'll look at that in the following slides. So reproductive isolation is this idea that you cannot mate with another species because of some isolation. And there are a few different types that we're going to go through on this slide as well as the next slide. So first of all, we can have ecological isolation. And what this means is the species are in completely different environments, so that is why they are not able to mate with one another. And the example given in your e-text is your Arctic fox and your desert fox. obviously two very different ecological niches, right? So they're in different environments. They're never going to encounter one another. That is ecological isolation. Next, we have temporal isolation. And essentially that means the organisms are just mating at different times of the year. So their mating is not going to align. They will not mate with one another. The example given in your e-text is the mating of the leopard frog. It mates in early spring. The bullfrog mates in early summer. So again, temporally, time-wise, their mating times do not align. Next, we have behavioral isolation. In behavioral isolation, we're seeing that different mating activities occur, and the organisms they're not attracted to the different mating displays. The example given here is the prairie chicken is not attracted to the mating display of ringneck pheasants. So they're not going to mate with one another. That mating display is done to attract mates, and if the organism isn't attracted to that, you're not going to have mating occur. The last type of reproductive isolation on this slide is mechanical isolation. So this is just like what it sounds. You have incompatible mating organs. So different types of pollinators is a good example of this. Over here you can see the hummingbird. The plants that are pollinated by the hummingbird do not receive pollination from plants that are pollinated by the black bee, right? They prefer different types of flowers. The hummingbird likes, these kind of tubular flowers, whereas bees tend to be less selective in terms of the flowers that they pollinate. So we'll look at a few more examples of reproductive isolation on the next slide. Continuing our discussion here of reproductive isolation, we also have gametic isolation. So what this means is the gametes cannot unite. So they can't unite and form a zygote, so reproduction cannot occur. So this would be an example of, really simple, say a cat and a dog. The sperm from a cat and the egg from a dog, when they unite, they will not form a zygote, right? We also have hybrid inviability. So this is where two organisms actually can mate, but when they do so, the zygote doesn't survive. And the example given in your textbook, I don't quite agree with. So they're showing that a goat and a sheep can mate, but that the zygote doesn't survive. However, there is something called a GEEP, G-E-E-P. Go ahead and look it up. So it actually does survive. So this is a very poor example of hybrid inviability, and I just wanted to point that out to you because your e-text kind of has that incorrect. The last type of reproductive isolation is hybrid infertility. This is where the zygote does survive. So you have this hybrid, it survives, but when it gets to the age that it is reproductively active, they can't reproduce. So a good example of this is when you take zebras and horses. They're different species, but their hybrid offspring that they produce, zebroids, they can't produce fertile offspring of their own. So these are all the different types of reproductive isolation that can occur. The last thing I want to talk about in this lecture is how exactly do we get new species? So we have something occurring known as speciation. This is the genetic divergence of populations, leading over time to reproductive isolation, and then after reproductive isolation, which we talked about on the following slide, or the previous slides, we have the formation of a new species. So new species may form when a strong barrier to gene flow occurs between the populations. And a really good example of this is, of course, Darwin's finches. So the first species of finch actually originated on the mainland of South America. So up here, mainland South America, kind of Ecuador region, right? And they spread down to the Galapagos archipelago. It's a series of islands that is actually off the coast of Ecuador. I don't know if you've ever been there. That's definitely on my bucket list to go to the Galapagos Islands. So as that original finch species moved from island to island, individuals were physically separated from one another by, of course, the ocean that was between each of the islands. So these separated populations then evolved in isolation from one another. And ultimately, the separated populations actually evolved such that they could not interbreed, which is pretty crazy, right? They became that genetically distinct, therefore becoming what we term now distinct finch species. So there are at least 13 finch species that actually diverged from that one original South American finch species. So you can see that here. These are the different islands in the Galapagos archipelago. So you have the South American ancestor. This is your blue-black grassquit. So that's the type of finch that it is. Then we have some that are now flower nectar eaters. They have a long tubular beak. That's the case of the cactus groundfinch. There are some that are seed eaters that have a strong cone-shaped beak, like the large groundfinch. And we have insect eaters that have this thin pointed beak, like the mangrove finch. So you've probably seen that famous original sketch or a copy of that showing the different beak shapes of these various finches. So there are actually 13 different species that all came from this original finch species that was on the South American, or it came from South America and then came down to the Galapagos Island. So pretty much what we see with this speciation is they were reproductively isolated from one another. They became adapted to the environment that they were in, and these new species formed. You may have also heard of something similar occurring with Hawaiian fish compared to very similar fish found in the Indo-Pacific, right? Hawaii is very isolated from everywhere else, right? And it's pretty rare. It would take like a hurricane or something else big in order to have reproduction occurring between Hawaiian fish species and those over in the Indo-Pacific, right? So that's why we have very distinct, what we call endemic fish species, so only found here in the Hawaiian Islands and found nowhere else in the world, because again, we have this reproductive isolation because Hawaii is so isolated from other regions. So that is all that I have for this lecture on Chapter 14. Make sure, if you haven't already, that you read Chapter 14 in your e-text. Also, make sure to complete the Chapter 14 adaptive quiz on Achieve. And if you have not already, make sure to at least plan your service learnings. I would hope that you would have completed at least one of your service learnings by now. If not, make sure you plan that out. You have to have them completed before finals week, right? So make sure you have it planned out. If you're getting stuck in terms of opportunities, email me and let me know. So that is all that I have for today. Mahalo for watching.